Hello, in this video I'm going to talk about cells. I'm going to start off by giving a brief introduction to cells, and then I'm mostly going to talk about the cell membrane. In this picture here we see a human cell called a macrophage, shown in purple, eating bacterial cells, shown in green. So first of all, how many cells are in the human body? Well, turns out no one can really agree. Trillions is kind of what they're saying. There's a lot. What's kind of scary in a way is that we have more bacterial cells in our body than we have human cells. So that just freaks me out just a little bit. But looking at the human cells in our body, right, we're going to talk about those in this particular topic. Cells are small, right? Most of them can't be seen without a microscope, right? So we measure them often in units called micrometers. And cells come in different sizes. You know, a typical cell size might be around 20 micrometers. A red blood cell is around 10, right? But some cells are rather large. For example, we have some neurons that are very, very, very long and run down like the whole length of your leg, for example. Or muscle cells that are very long too. Structure is important to the function of a cell. You know, going back to the muscle cell that I mentioned, you know, muscle cells are very long and thin, and that helps them to function. Whereas red blood cells have a very unique shape, right? They kind of look like a donut without a hole in the middle, and that's really important for their function of carrying oxygen. So even though we have lots of cells in our body, they look different and they act different too. Now, all human cells have certain structures in common. Okay, so these are things that we're going to find in all the cells in our body. So all human cells have DNA found inside of a nucleus. Right? DNA is the molecule of heredity. It's what's passed on from parent to offspring, and it contains the code to make proteins. All human cells also have a cytoplasm that contains organelles. Cytoplasm is usually described as a sort of semi-fluid matrix. I kind of think of it as like badly made jello. Okay, it's not quite the solid jello-y kind of texture, but it's not a liquid either, it's somewhere in between. And it has chunks in it, right, called organelles. The organelles are small little functioning units that we find inside the cell. Each organelle has a specific job that it does. Right? The watery part of the cytoplasm is called cytosol, or intracellular fluid. All human cells also have a cell membrane, also known as the plasma membrane, and that's the outer layer of the cell. And all human cells have something called the cytoskeleton, and this is a protein framework that we find inside of the cell that supports the cell and helps the cell to move and move materials around inside the cell. Now, when we look at all the cells in the human body, we can roughly separate them into two categories: germ cells and somatic cells. Now, germ cells doesn't mean like, ooh, germs here. Germ as in germinate, as in important for reproduction. Okay, so germ cells are the sex cells or gametes, and they include the sperm in the male and eggs in the female. All the other cells in the human body are somatic cells. The term soma, the prefix soma, means body. So the somatic cells are pretty much all the cells in your body that aren't the germ cells. So this would include the cells in your skin, the cells in your liver, your blood cells, that sort of thing. Even the cells that make up the wall of like the ovaries and testes. Germ cells and somatic cells are different in terms of how many chromosomes they contain. Germ cells have half the number of chromosomes compared to somatic cells. So germ cells in the human contain 23 chromosomes, whereas somatic cells contain 46. We use the term haploid to describe the half chromosome status of a germ cell, and we use the term diploid to refer to the fact that somatic cells have 2 of every chromosome. Here's a picture of a typical human cell, and we can see lots of different organelles inside the cell, right? Like Golgi, mitochondria, the nucleus, the endoplasmic reticulum. And in the background you see a lot of like strings and ropes and sticks and stuff. That's representing the cytoskeleton. And then of course you have a cell membrane, that outer barrier to the cell. That's what I want to talk about now, is the cell membrane or the plasma membrane. Now the plasma membrane or cell membrane is a phospholipid bilayer that acts as a barrier between a cell and its environment. So we talked about phospholipids in chemistry. Phospholipids are a type of lipid, and they were unique because they had both hydrophilic and hydrophobic properties, and we will review that in just a little bit. But these phospholipids create a double layer barrier that keeps stuff inside the cell and keeps stuff outside the cell. This barrier is protective, so it prevents bad things from getting into the cell and it prevents good things from getting out of the cell. But the cell membrane barrier is also important for things like transporting stuff into and out of the cell. It's also important for moving cells, because when a cell wants to move, the cell membrane has to move. The cell membrane is also important for communication, because sometimes we're going to use receptors in the cell membrane to receive messages and communicate with the cell. And there are many other functions of a cell membrane. What we're going to do right now is take a look at the structure of the cell membrane. So in this picture here, we can see a cross-section through a cell membrane. Towards the top of the screen, we're looking outside the cell, and towards the bottom of the screen, we're looking inside the cell. And the cell membrane is mostly made of the phospholipid bilayer, which is shown with the gold little squiggles and circles. Right? Those are the phospholipids. But you notice that the cell membrane is not made purely of a phospholipid bilayer. There's other stuff in there too. Right? Most notably are those big purple blobs, and those are proteins. Right? And then there's those smaller blue things, that's cholesterol. And then if you look on the outside of the cell membrane, you see stuff that looks like plants growing. Right? Those are carbohydrates, and those are also an important part to a cell membrane. So most of the cell membrane is made of phospholipids. The cholesterol that's there is there to help keep the membrane fluid, but not too fluid. So by taking in or removing cholesterol from the cell membrane, we can change how fluid or solid the cell membrane is, and that's important as temperatures change. The carbohydrates that are there are mostly on the external surface of the cell, and they're mostly there for identification purposes. It's really important for our defensive cells to be able to identify self cells as being self and then not killing those, right? So those carbohydrates help with that process. But then there's also a lot of proteins in the cell membrane too. And there are lots of different functions for the proteins in the cell membrane. So here we can see a series of pictures that describe some of the different functions that a protein in a cell membrane might have. In all of these pictures, the blue side on the left is supposed to be extracellular, and towards the right is intracellular. So in the first panel, we see a protein that's helping to transport substances into the cell. Next, we see a protein that's acting as an enzyme, right? It's changing diamonds into circles, but this could be converting, a carbohydrate into glucose. And then we see an example of a protein acting as a cell surface receptor. So this protein is receiving signals and then relaying that signal into the cell. Right? So this could be like a hormone or neurotransmitter that is sending a message to the cell. And then we see a protein with carbohydrates attached. So the carbohydrates are mostly for cell identification purposes. So this protein is helping with that process too. And then we see proteins interacting with each other to kind of hold cell membranes together. And then we see a protein attached to the cytoskeleton. And this could possibly help with the movement of the cell. So phospholipid bilayers are the primary component of cell membranes. So I want to talk a little bit more about those. But I also wanted to mention that phospholipid bilayers are also important to the structure to many organelles. So not only do they form that main component to the cell membrane, but these phospholipid bilayers form the majority of the organelles that we find inside of cells. Mitochondria, the nucleus, the Golgi apparatus, and the endoplasmic reticulum are all examples of organelles that are composed of a phospholipid bilayer. So let's review the structure of a phospholipid. Right? A phospholipid has two fatty acid chains. Okay? And these fatty acid chains are long chains of carbons and hydrogens. Okay? And those fatty acid chains are attached to a central group, and then that central group is also attached to a phosphate group. So that's where the term phospholipid comes from, is the fact that the phospholipid has phosphate. Phosphate contains the elements phosphorus and oxygen. Okay. The phosphate group has a charge to it. It has a negative charge to it. And so the phosphate group is very hydrophilic because things with charge love water. So the phosphate group of the phospholipid loves water. But those fatty acid chains that are made of carbons and hydrogens, those are extremely hydrophobic. They do not like water. So here we have in one molecule both a hydrophilic and a hydrophobic portion. Okay? The term for this is amphipathic. That's both hydrophilic and hydrophobic. And this is really important for the function of a phospholipid. And we'll look at that in just a minute. But I wanted to mention that we call the portion of the phospholipid that contains the phosphate group, we call that the head, right? So we say the head is hydrophilic. And the portion of the phospholipid that contains the fatty acid chains, we call that the tail, right? So the hydrophobic tail. When you put phospholipids in a watery environment, like we have in the body, phospholipids naturally form bilayers. What happens is the hydrophobic fatty acid tails face one another because like mixes with like, so the hydrophobic tails of one layer mix with the hydrophobic tails of the other layer. And then the hydrophilic heads face towards the watery environment. So in this picture here, we see a phospholipid bilayer. You'll notice that the fatty acid tails are facing one another and the hydrophilic heads are facing outward. But where's the water supposed to be in this picture? See if you can take a minute and figure that out. If you said the water was supposed to be here and here, you were correct. So phospholipid bilayers are a semi-permeable barrier. You can also say that a phospholipid bilayer is a selectively permeable barrier. So it's a barrier to some things, but not to others. In order to understand this, what we need to realize is that the majority of the phospholipid bilayer is hydrophobic. So let's take a look at that. Let's measure the width of our phospholipid bilayer. Right? And we'll say it's 10 nanometers. Now let's measure the portion of the phospholipid bilayer that is hydrophobic. And we'll say it's about 7 nanometers. Right? So basically 70% of the phospholipid bilayer is hydrophobic. And this is significant because like mixes with like. Most hydrophilic substances are not going to be able to cross the phospholipid bilayer because they will be repelled by that hydrophobic area in the middle of the phospholipid bilayer. So basically, if you want to get a hydrophobic substance across a phospholipid bilayer, it's going to need help of some kind. So what things can cross a phospholipid bilayer? Well, since most of the bilayer is hydrophobic, the real question is what things can cross the hydrophobic portion of the phospholipid bilayer. So here are some general rules for crossing phospholipid bilayers unaided. I say unaided because later on we'll look at what happens when we help things across the phospholipid bilayer using proteins. But general rules for crossing a phospholipid bilayer unaided. Number 1, you can cross if you are hydrophobic, no matter what your size is. And that's because you are basically made of the same stuff as the phospholipid bilayer. Number 2, you can cross just as long as you're not an ion. Right? Ions, no matter how big or how small, carry charge. And they're very hydrophilic, and they can't cross the hydrophobic portion of the phospholipid bilayer. And you can cross if you're not hydrophilic, which kind of sounds a lot like number 1. Number 1 says you can cross if you are hydrophobic, and number 3 says you can cross if you are not hydrophilic. Right? So it's kind of a little bit of overkill here, but it's a true statement. Hydrophilic substances have a really hard time crossing the hydrophobic portion of the phospholipid bilayer. The one exception to this is water. Water is, of course, hydrophilic, but because it's so small, it can kind of sneak across the phospholipid bilayer. So let's take a look at some examples. Right? Here we'll say this little pyramid represents oxygen gas. And oxygen gas is hydrophobic. Right? So it is able to slip right across the phospholipid bilayer. Ooh, this thing's pretty big, right? Way bigger than the oxygen gas. Let's say this is a steroid, like maybe estrogen or testosterone, right? So it is a lipid, right? Lipids are hydrophobic, right? So remember, the rule is you can cross if you're hydrophobic, no matter what your size is. So this particular molecule can cross because steroids are hydrophobic. We'll say this is glucose, a carbohydrate. It is hydrophilic. Now remember, the rule is you may cross just as long as you're not hydrophilic, so this thing's not going to be able to cross. Right? It's too big, it's too hydrophilic. It can't cross unaided. Ooh, so we have a little plus sign here. We'll say this represents a cation, a positively charged ion. Now remember, if you're an ion, you cannot cross that hydrophobic interior, right? You cannot cross the phospholipid bilayer unaided. See if you can take a minute and figure out which of the following items can cross a phospholipid bilayer unaided. So go ahead and pause the video and try to determine whether or not each of the following things can cross. All right, so let's take a look at number one, a magnesium ion. So a magnesium ion is an ion, so it carries charge. And anything with charge cannot cross the phospholipid bilayer unaided. A phosphate group, which also contains charge, cannot cross the phospholipid bilayer unaided. Cortisol is a steroid hormone. It's kind of like your stress hormone. Because it's made of a steroid, a lipid, it is able to cross the phospholipid bilayer unaided because it's hydrophobic. Glucose. Glucose is hydrophilic and cannot cross the phospholipid bilayer. Hydrogen ions are ions which carry charge and they cannot cross the phospholipid bilayer. Water is hydrophilic, but because it's so small, it does occasionally sneak across the phospholipid bilayer. Amino acids. These are the subunits that make up protein. Proteins and their subunits are hydrophilic, and they cannot cross the phospholipid bilayer. And last is a triglyceride. A triglyceride is a type of lipid, and it's hydrophobic, so it can cross the phospholipid bilayer.